Our first speaker is Dr. Alfredo Torres. Dr. Torres earned undergraduate degrees in chemistry, pharmacology, and biology, from also a master's degree in microbiology from the Benemérita Universidad Autónoma de Puebla. He went on to earn a PhD in molecular biology from UT Austin, and then completed a postdoctoral fellowship at the University of Maryland School of Medicine. Dr. Torres is currently the Herman Barnett Distinguished Professor in Microbiology and Immunology, and Associate Provost for Faculty Affairs and Diversity, Equity, and Inclusion at the University of Texas Medical Branch. He's a peer-recognized bacteriologist, vaccinologist with extensive experience in the study of bacterial pathogens causing diarrheal disease, such as pathogenic Escherichia coli and Shigella, as well as biodefense-related organisms, such as Burkholderia species. Dr. Torres has published more than 140 peer-reviewed articles, 15 book chapters, and 2 books on topics related to microbial pathogenesis, food safety, therapeutics, and vaccine development. In education, he's involved in teaching and mentoring a diverse group of trainees at local, national, and international levels, as well as being involved in the support and development of faculty at UTMB and in Latin America. He's been active in the American Biomedical Research Conference for Minoritized Scientists for many years. ABRCAMS is the largest national undergraduate research conference in the US, and St. Edward's students have been presenting there since 2007. Dr. Torres will be sharing his work with the art of making vaccines. Welcome to the Hilltop, Dr. Torres.
Hello, hello, can you hear me? Good. There are seats here at front, so you want to come in. Thank you, Trish. Thank you. Well, I'm deeply, deeply honored to be here. Thank you for the invitation, and I know that it's going to be tough to entertain you, being the first talk, 9 a.m. in the morning, and tell you about basic science. So what I decided to do, and I'm very loud, so I'm going to put it down, so you probably can hear me better. Is that okay on the back? Yes, okay. So I'm going to combine 3 things. One is my love of art, combined with we're celebrating the Hispanic Heritage Month. So one of my favorite artists of all time is the woman behind me, Frida Kahlo. So I'm going to combine that, how I combine the art of the life of Frida Kahlo with my experience with vaccines. And the whole idea of the whole talk, if you can get one thing, is I'm going to try to convince you that all the courses that you have to take in basic science are going to actually help you to be a vaccinology like me, okay? The final thing I'm going to say, I'm going to shut up about the things I'm going to convince you, is I do not take questions from faculty and senior people attending here. The first talk, or first question, I'm sorry, has to come from students. Is that a deal? Okay, let's go. So I'm going to tell you a little bit about the art of vaccines. And let me see if I can get here. Okay, so vaccines are like a canvas. Basically, when we are asked to develop a vaccine against a pathogen, you name it, could be a virus, a bacteria, a parasite, your favorite pathogen, this is what would end up. This is what normally we know, probably nothing about the pathogen. If we talk about SARS-CoV-2, we didn't know anything about it. So this is what we have to do. And at the end, we have to come up with a beautiful painting about what we can develop as a vaccine. So from a white canvas to a masterpiece, it can take you years, or like in the case of SARS-CoV-2, only took 6 to 8 months to develop a vaccine. It's how fast can we do. Do you know who this guy is? Have you heard about Edward Jenner? He's the first guy that developed vaccines, right? So actually, you can go to a museum and see a painting of Edward Jenner in a museum. Let's see. This is vaccination by Ernest Board. So how about Frida? What is Frida? So probably you saw a movie with my other favorite artist, Salma Hayek. It doesn't tell you about the story of Frida Kahlo, but the thing I want to tell you about Frida Kahlo is she was a woman of a lot of talent. She was one of the first female artist in Mexico that was recognized. And the way she expressed herself is portraits. And she was trying to express the pain and the suffering and the environment that she was working in or growing up or developing. One of the things that probably you heard about Frida Kahlo, she was in an accident. She suffered from this accident for many, many years, and the constant pain actually caused the changes in the art that she was expressing. So this is, she got married to a famous painter, a muralist in Mexico, Diego Rivera, and that's what she was. So you can go from Frida Kahlo, the early stages of the disease, to Frida Kahlo after all this, suffering and the pain. So if you translate this to vaccines, you can say that sometimes you have Fridas because you have some knowledge, and eventually you end up with Kahlos, that you try things and they don't work. So I'm going to tell you about 2 bacteria that we study in my lab. I can bet you never heard about this bacteria, and that's why I want to introduce you to them. One of them is Burkholderia malleae, that's the species name, and the other one is Burkholderia pseudomallei. Why are they important? Well, for those taking, I hope anybody has taken a microbiology class. They tell you about gram-negative bacteria, gram-positive bacteria. These are 2 gram-negative bacteria. That's how they are stained when you do it in the lab. They cause 2 diseases, glanders and myelodidosis. Again, probably you never heard about these diseases. And the interesting thing about is this bacteria can actually enter our cells. And the bacteria can actually grow inside of our cells, replicate, and cause disease once they're inside of our cells. So that's one of the characteristics of this bacteria. Okay, so that's what we know. It's a gram-negative, non-motile bacteria, and can survive in our cells. That's what we knew about this bacteria. Why do we got interested? Well, in my laboratory, we work in the Galveston National Laboratory, where we work with all these different pathogens that can cause disease naturally or man-made. One of these diseases, Burkholderia malleae, that causes glanders, actually is the only pathogen that actually has been used as a bioweapon. It was used in World War I and also in World War II. So the people that developed this bacteria and they modified this bacteria, they actually released the bacteria, got the horses infected and also the soldiers infected. They just waited, people got infected, and then they attacked them. So that was the first bioweapon. So the government here in the United States, the Department of Defense, has always been interested to develop vaccines against this pathogen. The other pathogen is called Burkholderia pseudomallei. It's very similar to malleae, but this one is a pathogen that you can actually find in the environment. If you travel to tropical areas around the world, particularly Thailand, Myanmar, Northern Australia, you can actually get infected when you go into the field, to the rice fields, and then the bacteria can enter through your skin naturally, or if you go to Australia and you get caught in a monsoon, you can actually inhale the bacteria. Or the other thing that you can actually acquire this bacteria is through the gastrointestinal tract. So food or water contaminated with this bacteria can actually infect you. So why are we studying this bacteria? Well, it turns out that this year we actually found, the CDC actually found this bacteria in Mississippi. 2 people that got infected died from the infection, and they actually found it for the first time in the soil of Mississippi. We have had 2 cases here in Texas. So the bacteria is here around us. It doesn't cause a lot of infections, but with global, the climate change and all these changes that we are seeing, potentially this bacteria can adapt and start causing more infections. So it's better to be prepared. So that's all we know about this bacteria. So now develop a vaccine. What do you do? So that's the challenge for you. That's what basically the government told us in my lab. So we have these 2 bacteria. This is the information that we have. We want a vaccine developed in 2 years. Here's the money. Figure it out. So how do we do it? Well, we go to Frida. And we asked Frida, what can we do? So Frida is wearing this traditional dress. Represents the southern part of Mexico. It's in Oaxaca. And the interesting thing is basically she was trying to express the beauty of Mexico. And she's wearing this dress and beauty, showing, all this dress outside of her and the beauty that is giving her because wearing this particular dress. So bacteria also express different things on the surface that we can actually study. Not like Frida, not as beautiful as Frida, but these bacteria can actually use these surface structures and we can understand them. And we can use them for developing vaccines. So one thing that we did is, again, going to the basic science, and if you have ever taken genetics, I hope anybody has taken any genetics class, we can actually create mutations. We can use that information to create mutations. So inactivate genes in this bacteria and make it less virulent, so unable to cause disease. So we create a mutation in a process that is natural for this bacteria. So this bacteria, we know that iron is required for any living thing to survive. If we don't have iron, we die. So the same with bacteria. If the bacteria doesn't have iron, the bacteria cannot replicate, cannot multiply, and cannot cause infection. So once the bacteria goes inside our cells, the iron concentration is very low. So the bacteria has a hard time surviving. So it has to have mechanisms to sequester this iron from the cells and survive. So we ask the question, can we inactivate one of these genes that now they make the bacteria unable to use iron and then unable to cause disease and use that as a vaccine? So we did that. So we inactivate the ability to use iron. The other thing that we inactivate, we realize, is that this bacteria is able to, once infect one of our cells, the bacteria can move from cell to cell, infecting this cell and then moving to the next cell without leaving the intracellular environment. So we figured out that if we inactivate another gene that makes the bacteria unable to move from cell to cell, we trap the bacteria in one cell, and then we let the immune system to attack the bacteria and actually produce an immune response that protects against the infection. So we inactivate this. So basically what we did is inactivate two genes. Again, the basic genetics, we inactivate two genes. And then unfortunately, we have to use an animal model to test whether this vaccine works. So we use this mutated bacteria to vaccinate these animals. And then we challenge the animals with a lethal dose of the bacteria, the wild-type bacteria, to see if the animals survive or the animals die. Are we all with me? Are we following me? Good, super. But remember, you have to ask me questions. So, and then we can do aerosol challenge, and we can check, if you have taken immunology class, we look for antibodies or for cells that are actually involved in the immune system responses, and then we look for those. We can actually quantify whether we have increase of those cells once we vaccinate. We have increase of antibodies if we vaccinate. And then we look for safety and efficacy. We don't want the animals to get sick with the vaccine, because eventually we are going to be using this in humans. So basically what we do is we vaccinate 3 doses of the vaccine to these animals, and then we challenge with a lethal dose, and we take the organs and examine whether the bacteria can actually move to the different organs and whether the wild-type bacteria can survive. We can do predictions. So once we vaccinate these animals, we can take sera, blood, and then take sera from these animals and quantify how much antibodies are being made. And again, basic immunology. If you see an increase in antibodies, you can predict whether these animals are going to survive to the infection. If these animals do not make enough antibodies, we can predict that these animals will not survive the infection. So that's what we did. We vaccinated animals, with different doses of the vaccine. And we saw with this dose of the vaccine, the animals made 20,000 units of antibodies. But if we challenge with this, immunize with this vaccine dose, we got 42,000. So our prediction was that these animals will not be protected or slightly protected, and these animals will be protected. So we did the experiment, and then the animals that were not vaccinated, they die from the infection. the animals with a low dose of the vaccine, 70% of them survived. 100% of the animals that got the dose survived. So we are going in the right direction, right? So it seems like the vaccine is working. So can we define the correlates of protection? So can we understand better how these immune responses in our animals are producing the protection? Well, people in my lab went to Frida again and asked Frida, can we try to understand or figure it out, how can we do this? So Frida paintings evolved. Before it was just trying to show the beauty of Mexico. And eventually Frida started asking the basic questions about her own humanity and how she saw herself in the Mexican culture. So at one point during her career, she thought that Frida was two Fridas, not just one Frida. The Frida that it was interacting with people in Europe, and the Frida that was a true Mexican Frida. That was that interaction of, and again, I don't have the time to explain why these things were happening. If you can read a book about Frida, it's exciting and interesting, but basically she was debating about these two cultures that were happening in Mexico. So she asked this question. So basically we say, can we actually figure it out, the different components of these two Fridas in our vaccine? And that's what we did. Again, we do these challenges where we can get 100% protection of our animals with the vaccine. The animals that are not vaccinated, they die. And then we look for the bacteria in the different organs, and we saw that very few bacteria survive. It's not perfect. We got these dots represent some animals that have some bacteria, but I guess we were able to protect. And then we start looking for the production of antibodies. For those that have taken immunology, you know that there is different types of antibodies, and each one of these antibodies have different functions, right? The common one that they ask you to do is measure IgG, and then you can actually measure IgG. If there is an increase of IgG, indicates that you might get some protection due to the vaccine. And there is subtypes of IgG that we can actually measure, and we can predict whether these subtypes of IgGs are actually being induced, and that can predict that we can actually protect these animals. Turns out that our vaccine produces IgG, IgG1, and IgG2 increases, and that's how we figured out that the protection that we're seeing is due to these antibodies that were being produced. I'm trying to make it very simple. Obviously, there is a lot of thinking behind. And the other thing that we can actually measure is whether there is cells, not just the antibodies, but actually cells that can actually be induced due to the vaccine, and those cells are participating in the protection. So basic immunology class, the professor told you there is a humoral response, the antibodies, and the cellular response. And some vaccines are very good just inducing antibodies, and that's all you get. But some other vaccines, especially for intracellular pathogens, you have to have a humoral response and also a cellular response. And that's why it's so complicated to have vaccines against intracellular pathogens. The best example, tuberculosis. Tuberculosis is an intracellular pathogen, and it's very difficult to develop a vaccine against tuberculosis. And this is the reason. So basically what we have is we can actually measure different types of cells, CD4 and CD8. These are T cells. And you can see that if you deplete these cells, you get increased infection. Some of them, if you deplete CD8, nothing happens. So it seems like CD4s are critical for protection. Okay. So what I have told you so far is that we can protect these animals from the infection. We eliminate the wild-type infection, increase antibodies. We didn't see any damage obviously for the time being, I couldn't show you, but we didn't see any damage to the tissues. We didn't see inflammation. We saw CD4 T cells being induced, persistent, reduced, eliminated. It cannot revert to the wild type because we have created 2 mutations, and they cannot become wild type. And it's attenuated in immunocompromised mice. So that's what we went back to the Department of Defense and say, we have this vaccine, we're ready to go, ready to start testing it. And they told me, well, this is a bacteria. Some people might not want to get immunize with a bacteria because it might be dangerous. So go back to the drawing board and figure out a better vaccine that doesn't have the entire bacteria, but it only has one component or two components of the vaccine, and it still works. So here's the money, figure it out. So what do? I go back to Frida, right? So Frida, through the years, now she started having all these issues with health. You know, after the accident, she broke her back, and then she suffered a lot. And at the time, again, medicine was not as advanced as today, and she was really in constant pain. And you can actually see the progression of that pain in her paintings. One of these is this one in 1944, the broken spine, where she starts showing that, you know, she was suffering. But even in these paintings, you can look at her eyes, and there's people's doves of peace. This is my joke about pain and suffering. There is always the good thing about pain. There is always positive things about pain. So that was the pain that we were suffering at the moment that we spent all this time developing a vaccine, and they said, well, maybe that's not the right vaccine. So we know that a lot of people smarter than us develop vaccines, and they develop different techniques to actually develop these vaccines. So these are many pathogens, and probably you here in college, you came and they told you you have to get the meningococcal vaccine because you're going to be in dorms or you're going to be in these things, and they don't ask you. You have to get vaccinated, otherwise you don't get into the university. So many of these vaccines have actually been used, different techniques to develop, but there is other ones, preventable diseases, that use different techniques. So we use the same methodology that they use for the meningococcal vaccine. It's called reverse vaccinology. And I'm going to tell you that in a minute. So if you have ever taken a bioinformatics course, raise your hand if you have ever taken a bioinformatics course. Fantastic. Guess what? It's actually useful. It's actually useful. So I went back to my lab and I say, how can we use bioinformatics to figure it out what proteins that are expressed on the surface of the bacteria can actually work as a vaccine candidate or a component of a vaccine. So the very smart grad students in my laboratory, they say, well, let's do bioinformatics, figuring out few things about these bacteria. We're looking for proteins, and then proteins are structures that are on the surface of the bacteria that are conserved in Burkholderia, that are not present in any other organism. That was one. Two, that are on the surface, because once we get infected, the first thing that we see in a bacteria is the surface, right? So if we get antibodies against surface bacteria, we can actually prevent that this bacteria enters our cells. So that's what we got. And then we're looking for the proteins that have less than one transmembrane domain. If you have ever taken a biochemistry course and they told you that, oh, it's very easy to purify proteins, tell your professor that's a lie. If a protein is a protein that has multiple transmembrane domains, it's very difficult to get it out of the membrane, very difficult to purify. So our goal was to try to find proteins that have only one transmembrane domain that was very easy to purify, similarity to other antigens that have actually been discovered by other bacteria, similarity to non-adhesins, and again, those proteins that actually activate or induce the immune response. We can actually do that in a computer. You don't have to go to the lab. You can just be in the computer and type and use all these programs that can figure this out. So basically, my student Laura Muruato took the genes, the genome of this bacteria, and there's about 5,000 proteins. So that's a lot of proteins to figure that out. So she started using a bioinformatic approach. She looked for subcellular localization. She ended up with 160 proteins, physical chemical properties, 132 proteins, stability, 106 proteins. Oops, sorry. Antigen properties, 104. And I told her, when she showed me it has 100 proteins, say, well, try to purify 100 proteins. And she said, I'm quitting. So it's like, okay, let's try to rank this. So we took all these lists of 100 and we ranked them. We thought the best one to the worst one. And then we took the top 7, and then we start trying to purify and cloning this. So Very easy, right? So have you ever done a PCR reaction in your lab? Or have you ever learned about PCR? So you can amplify a gene, right? And then you can clone it. And then you can express the protein. Guess what? Burkholderia doesn't like PCRs. So Burkholderia, again, in the genome, when they teach you PCR, they told you have to amplify the gene, right? One of the steps. And they told you, well, it's very easy. Just do the annealing and get the gene, and you can amplify, and you can calculate what primers you use and all that. That's what they teach you, right? These bacteria, the genes have about 70 to 90% GC content. So when you try to do PCR, it's impossible. The annealing temperature is like 80 degrees. So it's impossible to amplify. Some of these proteins are very difficult. Sorry for those that do not have PCR experience, but this is basically a simple technique that we use to it. So we try to, amplify. These are the only ones, the ones in red, that were the only ones that were able to amplify and clone. And, these are the genes, and they are located on the surface, similar to what the bioinformatics predicted. And this is what we start working with. So we went back to Frida, and I say, what else can we do? So again, this is the latest stages of Frida's career. This is 1932, when she was in the Henry Ford Hospital, suffering from many health issues. You can read about it. Basically, she was in a lot of suffering, and she was trying to understand why her own existence as an artist and a person in Mexico was transcending, her humanity. And she was asking, well, there were different things in my life that has basically impacted my life. One of the things that we decide to do is take this example of different parts and ask the question, can we use our basic knowledge? So they told you that I have a background in chemistry, right? So I said, well, let's use my background in chemistry. Can we use basic chemistry to develop a vaccine to assemble these proteins that now we have with basic chemistry? So we learned that actually we can use gold nanoparticles, very, nanotechnology, use that gold nanoparticles to put proteins on top of these gold nanoparticles. So we use chemistry to put these linkers on the surface of these gold nanoparticles. And then we can assemble the protein of interest, plus we can put carbohydrates or other things on the surface. So basically what you end up having is a gold nanoparticle cover with these proteins. And you can use that as a vaccine. So I'm giving you a question for me. So I'm not going to tell you why, but if you're curious to know why do we use gold, right? So there's a question. So you can type it. There is a QR code over there. So basically, if you have done purification in the lab, you can actually purify proteins. You can purify the LPS or carbohydrates. And then you can do chemistry in the lab. We play with the chemistry. And then we assemble these gold nanoparticles. We put the linker. We can put the protein. And then we can engineer the lipopolysaccharide. This looked very good. Took us a year to figure out how to assemble this and how stable this nanovaccine is. And eventually, my student Daniel Tapia developed this vaccine. We can actually vaccinate these animals. And again, challenge with a little dose of the bacteria. And ask the question, can we actually develop an immune response that protects our animals? And we can get protection. So these are some of the examples of the proteins that we engineer with different combinations. And again, the first experiment, he came to my office and he said, Alfredo, it didn't really work very well. All the animals receiving different combinations, they all die similar to unvaccinated animals. But there was two of them that seemed to be promising. This one in blue, again, it doesn't matter what the name of the protein is. Two in blue that seems to be some protective. And then the one in green, it seems to give 90% protection. So we went back, re-engineer, play around with these proteins. And the next time we did it, now we have proteins that actually protect 80 to 90%. And if we combine the two proteins, now we have 100% protection with two proteins. So instead of having the whole bacteria, we can just use two proteins and we can still get protection. And this is, the numbers of bacteria that we can find in the different organs. So it's not perfect. We're perfecting this to try to eliminate the bacteria in the different organs. We can get, again, looking for antibodies. We can get antibodies against the different proteins. And we can also get antibodies against the lipopolysaccharide. Remember, we put the lipopolysaccharide and the protein. So we generate antibodies against those two components. So just to summarize and to finish my talk, I hope I give you an idea that, playing around with the basic knowledge that you guys are getting right now, you can actually apply it to very complicated questions that we have in vaccine development. And you can combine, I'm not an immunologist, I admit that. So I have to learn immunology as I was going with this vaccine. So now I think I know a little bit about immunology, because we can actually generate immune responses that are actually protective. And then we can actually start doing, we can actually start doing more complicated things to try to define what cells we have to induce with this vaccine to actually get protection. So that's a different talk that I can give you. But at the end of the game, these bacteria, like SARS-CoV-2 causing COVID-19, the virus in that case start mutating, right? And then the vaccine that was designed for type of SARS-CoV-2 or causing COVID-19 starts mutating, and the vaccine is not that effective anymore. So viruses, bacteria, parasites figured out that they're attacking them, and they try to find a way to go around. So these bacteria like that is very, very, I don't want to say smart, but finds the way to go around. So this is always in my lab, to remember that even want to drown my sorrows in liquor, but the dumb ones learn to swim. So bacteria seems to be doing the same thing. So we're now playing around, trying to combine and perfect our vaccines to try to prevent that they learn how to swim, these bacteria. This is the people in my lab, the super talented grad students, Jacob, Sarah, and Alex. I have two postdoctoral trainees, Itziar and Nitalia, visiting professor from Mexico. This is my lab manager, and this is the funding and collaborators. And as I told you, I do not take questions from faculty or invited guests. I take questions from students. Thank you very much for your time. Sorry, I put you on the spot, guys. So you have to.
If you want to ask your question on the microphone, you're welcome to do that. If you'd rather type it in, then you can type it in. So, yeah.
I'm not that intimidating, right? So you can ask me face to face.
The best compliment you can give a speaker is to ask a question. So if you don't want to offend him, you should.
Yes, please. Super. Why do you use the gold? Damn. Well, turns out that we actually use gold for some of medicines that we actually use for rheumatoid arthritis. It's well tolerated, and it's not that expensive to use. It's easy to engineer, and that's why we use it. It's tiny particles that we can use, so it's not expensive to do it. That's why. Yes, good.
The proteins that you ended up purifying, would those be considered recognizing those regularly, like on the outside of the bacteria?
Very smart question. So PAMS is basically the host can actually recognize these proteins. That's what happens. There is receptors in our cells that can actually recognize. The answer is no. The bacteria actually use those to try to avoid the immune system. So, but they are required for the bacteria to actually cause infection. So if we get antibodies that actually bind to those surface proteins, we can prevent the bacteria to actually go inside the cells. Because it's game over. The reason is not, the vaccines are not working 100%, is once the bacteria goes inside the cells, we cannot do much. So we have to attack the bacteria before it goes inside of our cells. Yes? Yeah.
You said you were discovering the bacteria go from one cell to another.
Mm-hmm.
Did you discover the machinery or the mechanism that they use to get past the bilayer?
Absolutely. So we did not discover the mechanism. We knew that the mechanism was occurring and somebody else discovered, and then we figured out if we inactivate that a particular gene from that system, we were trapping the bacteria inside the cell. So the bacteria was not dying. The bacteria was still able to infect, but it was unable to move from cell to cell. So that gave time for our immune system to actually, when the cell die, the bacteria will be attacked, and then we get immune response. Mm-hmm.
So how long have you been working on vaccines for Burkholderia?
10 years. 10 years.
So when you have this gold nanoparticle and you attach to the biolates and you have the protein, how do you characterize that the proteins are actually absorbed to the
That's a very good question. So we use NMR, we use electron microscopy to actually figure out whether we assemble. It's not an effective, I should not say effective. It's, the outcome when we do this is not great. Sometimes we get 30 to 40% efficiency. That was the word I was looking for, efficiency in this. So we still have to perfect. So I have Alexander is my new grad student. He has a chemistry background. So he quickly realized the linker I'm using, or we selected, it's not the perfect linker. So now he's testing different linkers. And then he increased solubility. With these longer linkers, we can actually put more proteins on the surface. So, in a year, hopefully we get a perfected vaccine. Yeah. Mm-hmm. Any other question? Yes? It's nothing unique about the bacteria. It seems, to be honest with you, the bacteria is a bacteria that you can find on the soil in endemic areas around the world, basically in tropical areas. So the bacteria accidentally can cause infection in humans. And as I always tell my students when I'm teaching a class, some of these bacteria really don't want to be inside of you. So the only reason sometimes they kill you is because they want to go out and go back to the environment. The best example is cholera. The people die from cholera, for this massive diarrhea, because the bacteria wants to go out to the environment. The bacteria really doesn't want to be inside of you. And in the process, unfortunately, sometimes we die from the infection. The same thing with this one. However, the bacteria has the ability to actually adapt very quickly. So there's cases that people get infected in Thailand or in Southeast Asia, and then they are treated, but the bacteria doesn't disappear. The bacteria stays there for years, and then the infection can actually reemerge 60 years later when the immune system goes down. So the bacteria finds the way to go to a particular organ. They like the spleen, they like the liver, and then stay there, dormant. So one part of my lab is trying to understand why the bacteria actually persist, and how can we eliminate that step. Yeah. You have any?
So it seems like in your experiments that the mice died within a couple of days.
Yes.
Fast. Do humans die as quickly?
No. The model is very aggressive. The model that we use is an aerosol model. Again, as I told you, and I was not lying to you, the Department of Defense was looking for a vaccine that can protect our soldiers from potential exposure. And the potential exposure that the soldiers can get is through aerosol, if they were using a bioweapon. Eventually now, we're looking for different routes of infection. As I told you, the bacteria can enter through your skin. If you go to a rice field and then you use protection, can actually go through your foot and then go through your skin, or you can actually eat it, ingest it. So we're trying to develop now vaccines that can actually protect subjects in endemic areas that can actually just accidentally get in contact with the bacteria. Mm-hmm. Yes, now you can ask me.
I was at a meeting the other day, and there were several older women that said they were going to refuse to get the COVID-19 vaccine.
Sure.
I could not understand that. Now, I am a product of the first polio vaccines. I've had all 4 of them. Measles vaccines. Every time my children signed up for getting a vaccine, I said, give it to me. I don't understand why people are hesitating to get this vaccine. I've had 4.
Can I answer a question with what I honestly think about it? let me ask, let me revert your question with another question for you. And you can tell that to your people, to friends that you know. Here in the room, let me ask the people in the room, have you have anybody in your family that suffer from cancer? A lot, right? Do you have any concerns when these people go for treatment? You send it for treatment, right? My mother right now is suffering from breast cancer, and she went through chemo and all these different treatments. Have you ever thought about the consequences of sending somebody to treatment? 100% of the treatment against cancer is toxic. And a lot of people die from the treatment. But we do not hesitate to send in our family members to get treated against cancer. Now, let me ask you. So when you get these vaccines, people think that they're going to die from the vaccine because a lot of rumors and a lot of things. Why cancer is treated one way? Why an infectious disease is treated the other way? Okay, let me ask you the following. There is actually vaccines that have been developed now against cancer. Can ask you these people, if there is a vaccine that prevent cancer or prevent Alzheimer's, there is a vaccine against Alzheimer's now, will you take it? I can bet you that those people will immediately change their mind and say, oh, of course I will take a cancer vaccine. Of course I will take an Alzheimer's vaccine. So why are you not taking a COVID-19 vaccine? It's misinformation. That's the answer I can give you. Sorry, I'm very passionate about this. That's why I was asking permission to.
So here's a question. When your vaccine becomes approved, who will likely get it? Will it be shared with countries like Thailand?
Absolutely. So big thing about vaccines is not that we cannot develop vaccines. We have the thing. I showed you that in a couple of years we were able to develop a vaccine that eventually is moving into preclinical trials and is safe and is effective. What is the bottleneck and the problem? The money to take the vaccine from the laboratory to actually being used in human use. So some of these countries might be able to fund a vaccine that can apply to their country, but if you want to develop a vaccine that is going to be widely distributed, pharmaceutical companies will not invest the money to develop these vaccines. So diarrhea can be prevented. We have developed a vaccine against diarrheal diseases. Nobody wants to take that vaccine in the pharmaceutical company because they do not generate money. And that's the reality. I'm not complaining, it's what we experience as developers of vaccines. Are we okay with time?
Yes.
Okay, I don't want to take your time. Yes, you have any question? No? Yes. Absolutely. So that's what we are doing real time now with vaccines against COVID-19. So you develop the vaccines. There's 3 phases that we have to go through. Probably the next speaker is going to tell a little bit about that. But just quickly, we go through hundreds of people to thousands of people. And then we look for different markers that identify whether it's safe and effective. And then how you react. And then you follow up these people. My family, my son, my wife and I were the first Latino in Galveston participating in a clinical trial with the Pfizer vaccine. So they were, we're excited because there's data that showed that Latinos do not participate in clinical trials. So we never know whether the vaccine is going to work for an ethnic group, but not for the other one, right? So it happens that maybe some ethnic group might not respond properly. So we went, and I'm still in the study. I have my phone, and every week I have to report whether I get infected with COVID, or I have any symptoms, or something is weird with me, and extra arm is coming out of me, something like that, and they keep following me. I'm still part of the study. So they follow me for 2 years, and then see if anything happened to me. And that's how we know that the vaccines are effective and safe. Yep. Yes?
You said how bacteria mutate so they could invade immune response.
Mhm.
So how could we detect genetic changes in bacteria so we could ensure the vaccine are effective enough to protect us?
Yeah, I can tell you that I picked the wrong organism to study because when you study viruses, and I'm not saying anything, I hope there's no virologists in the room. Viruses are very easy to follow because they have the genome is very small. They have 15 genes, and you can actually sequence these 15 genes over and over and over, and then figure out quickly detect the bacteria. This one has 5,000 genes. So to sequence all of them and then assemble the genome and figure out which mutation is causing that change is very difficult. So for bacteria, fortunately, the bacteria mutates very slowly. It's not like viruses that mutate faster, but it can happen. So they can become resistant to antibiotics, which this one is super resistant to antibiotics. They can change their ability to survive in different environments. Some of the isolates that we have identified are more virulent than others. For example, you can only put, and I mean it, one bacteria in an animal, and you can kill an animal in 3 days, one bacteria. So the genetic component inside of this bacteria is different, and we don't know much about it. So we're trying to understand as we go. Mm-hmm. Yes?
How is it determined when like a booster would be needed rather than just?
That's a fantastic question. The answer is we don't know. So the methodology that we use is we use a prime and 2 boosts, similar that we did for COVID, thinking that we just want to see if we can actually protect. Later on, we will figure out whether we can reduce the number of boosts or we might need more. It's a lot of trial and error in what we do. So yeah, we don't know. Yes, sir.
A friend of mine who had cancer was in the autoimmune clinical trials where basically he was on the verge of dying and wiped out his cancer except one little thing. Is that considered a vaccine? And why doesn't it work on all types of cancer?
No, it's not considered a vaccine. There is different treatments and therapeutics that are used. I know there is actually vaccines being tested right now in clinical trials that it will prevent that people develop a particular type of cancer, or what they do is actually once you start developing symptoms or they have signs of the cancer, can actually attack those cells and start attacking. So they're considered vaccines, but they are not really vaccines. They are immunotreatments, what you're seeing.
What I understand is that they take blood out and separate the T cells out, incubate them in the tin,
And then they put it back. So technically, it's not a vaccine. It's basically they are purifying and then they're enriching our own cells, and then they are sending it back, primed to attack what they want to do. So in that technical term will be, yeah, it's kind of a vaccine, but it's our own vaccine. Yeah. But that's the beauty now that we have. We can actually engineer or play with the cells, put them back.
really worked for him, and he was on the verge.
Absolutely, absolutely, absolutely. Yes, yes.
Was there any specific reason you used mice as your model organism? Like, was the pathology similar to that of humans? Did you not know that going into it, and that was like the cheapest option?
I should recruit you to my lab. So the closest model for us to test is non-human primates. Each animal costs $16,000 to do an experiment. So guess what? We have to figure out an animal that costs us $30 an animal, which is a mouse, and develop the vaccines in mice. We have done testing in non-human primates. The vaccine works, but it's a super, super expensive experiment. No, no. This bacteria can actually, with the right dose, can actually kill anything. That's. I'm sorry for saying that way, but that's what we experience. Yeah. Thank you. Thank you very much. Thank you.